And so now I'm going to go to the next talk. Now, Dr. Fenari will give us now a discussion on core one, on the study on the resistant mechanism. Take it away, Frank.
Great. Thank you for this opportunity for us to present our approach for discovery. I'm going to pick up on the point that Dr. Young made, which is that drugs and therapies for GBM patients don't lead to a durable cure. And so our goal as part of Core 1 is to identify the right targets and associated resistance mechanisms to overcome these reasons for upfront resistance or acquired resistance in GBM patients. So we have 4 aims in our project. Aim 1 is to determine the signaling mechanisms which mediate escape from a very prominent oncogene in glioblastoma known as delta EGFR. And I'll tell you a little bit about that in a couple of slides. So we're interested in determining how GBMs can actually escape the need for the expression and activity of this molecule. So those would represent resistance mechanisms. Secondly, we want to identify the genes which are actually providing this escape mechanism. In other words, making the tumors delta EGFR independent for growth. And then thirdly, now we're considering looking outside the tumor and considering the environment around the tumor, known as the microenvironment. And here we would like to know what contribution the microenvironment has in mediating resistance to therapies targeting delta EGFR. So we're trying to take a holistic approach to understanding how tumors can escape the need or the function for this oncogene which drives their proliferation. And then lastly, what we learn from the first 3 aims will be, we'll use that to generate therapeutic or models that we can then apply to projects 2 and projects 3 in our consortium. Okay, so the receptor or epidermal growth factor receptor that we study is a receptor tyrosine kinase. This is a molecule that sits on the cell membrane of the tumor cells and provides growth signals to the tumor cells. So it helps the tumor cell to proliferate quicker than normal cells. So EGFR is found to be amplified or overexpressed in about 50% of all GBMs that we look at. So 50% of those tumors have a lot of expression of this oncogene. And then when we look a little closer, we actually see the occurrence of a mutated form of this receptor, which we refer to as delta EGFR. Hopefully you can see my cursor on the screen. This molecule, delta EGFR, is a mutant form of the molecule here shown on the left. Now, over the years, Dr. Cavaney and I have ascribed a number of functions to this molecule, delta EGFR, which, as I said, is expressed in about 1/4 of all gliomas. It does many things to the tumor cell, but I'll just highlight 2 of them. One is if you express it in a tumor cell and you engineer these cells into the brains of mice to model the tumor, you can see here in this brown area that it has a very big potential to enhance the tumor growth. And that's relative to the controls in the other panels. The controls are tumor cells which don't have that receptor or have other versions of EGFR. So this mutated receptor, delta EGFR, conveys enhanced tumorigenicity. And over here on the right, it also conveys therapeutic resistance. So when cells express this delta EGFR molecule and you treat them with chemo drugs such as cisplatin, the cells tend to survive in the presence of these chemotherapeutics. So given this information, many labs around the world have been developing small molecules and antibodies to target delta EGFR and wild-type EGFR. When you look across tumor types, EGFR is involved in several types of cancers, non-small cell lung cancer, colon cancer, to name a few. And there, these inhibitors, these antibodies, have yielded some efficacy. However, in brain tumors, this is clearly not the case. So in GBM, as shown here, we have two scenarios. We have a responder and a non-responder. The responders here to an EGF receptor inhibitor, you can see that there's actually shrinkage of the tumor mass on this MRI. Whereas the non-responders, you can see on therapy, the tumor actually has gotten much bigger. And through the elegant work of Ingo Mellenhoff, who you'll hear about after my presentation, he showed a number of years ago that the difference between a responder and a non-responder was in large part due to the presence of another gene in these tumor cells known as P10. So P10 is involved in controlling some of the signaling output of delta EGFR. And when the patients have wild-type P10, as shown in this brown part of the graph, they tend to respond to drug. Whereas those patients who have mutated P10, they fall into the group known as non-responders. But if you look closely at this, it's just not a cure. So even when you have wild-type P10, the time to progression is about 10 months, but nonetheless, the patients relapse on therapy. So we are very interested in determining why these patients relapse on therapy. They have the receptor being expressed, they have wild-type P10, but after a while, they no longer respond. And so that will be, I'll tell you a little bit about that. So that is one of our projects that we're trying to determine how the cells become independent of growth and acquire resistance to EGF receptor inhibitors. And so we've taken two approaches to address this. We have a genetic system that we've established in the lab, which is what I'll tell you about. Here, what we've done is to take a glioma cell line, engineer in delta EGFR, where we could turn it on and turn it off using some genetic tricks. And when the receptor is on, you can see that the cells make very robust tumors. This is a tumor assay in mice. So the blue line indicates that the tumors are growing very rapidly, and they're also expressing delta EGFR. However, when we turn off the receptor through our genetic manipulation, the tumors go through a mild regression, as shown here, and then over time, they actually resume growth, as shown here. Much like what happens in patients. This is a period of stasis when the receptor is been turned off, and then the tumors find a way to resume growth in the absence of the expression of this oncogene known as delta EGFR, shown below. The other system, which unfortunately there's not enough time to tell you about today, is our pharmacological system. Here on the right, we've been using mouse astrocytes, which express delta EGFR, and when they're engineered or implanted into the brains of nude mice, they form gliomas quite well. These astrocytes are very sensitive to EGF receptor inhibitors, and we've now been able to generate EGF receptor inhibitor resistant mouse astrocytes, much like what occurs in the clinical scenario. So I'm just going to tell you a little bit more about our system on the left. So the first thing that we've done is to try to profile these, what we call escaper cells. These are cells which grow in the absence of delta EGFR. And that was simply to look at their growth kinetics as well as their ability to undergo cell death. And what we found was when you turn off the receptor, the immediate response of the tumors was that they stopped growing, as indicated by this decrease here in a marker we call Ki67. And there was a mild increase in cell death, as shown here by this bar on the right. However, when the cells became clearly independent of delta EGFR and resumed growth, what we found was that they really don't increase their proliferative nature, but they decrease their ability to die. So what we're seeing here is that the tumor cells have found a way to overcome cell death and continue, and therefore they're able to proliferate. So with this, we reasoned that perhaps the tumor cells were actually changing how they transmit a growth signal to the tumor. And what we found was that they switch in a pathway known as MAP kinase. So if you look here on the left, this is what we call a Western blot analysis. We have the delta EGFR dependent tumor cells, which show that they have very low levels of these MAP kinase proteins. However, the delta EGFR independent escapers turn on very high levels of both of these proteins as shown here. And also, if you look at individual cells as shown here with a stain that detects these proteins, you can see that they're present in the escapers, but not really present in the delta EGFR dependent tumors. So this would suggest that the delta EGFR independent tumors are now sensitive to inhibitors that target the MAP kinase pathway, whereas the delta EGFR dependent tumors are not. And that proved exactly the case. That's shown here. So if you look at the green bars, these are what we call the delta EGFR independent tumors. When they're treated with an inhibitor to the MAP kinase pathway, they undergo a decrease in cell proliferation. Whereas the red bars, these are the delta EGFR dependent tumors, they pretty much ignore the drug that we use here that blocks the MAP kinase pathway. And we went on to further show that the delta EGFR independent tumors have rewired in such a way that they drive MAP kinase signaling in the absence of delta EGFR expression, as shown here. Okay, and so as my last slide, in addition to looking at how the cells differ in their signaling potential, which would be targetable molecules, we've also looked at how the cells differ in the genes that they express. And so to do this, we did what's known as a transcription array. This is a snapshot look at all the genes which are expressed in a particular type of cell. And we generate these heat map profiles as shown here. Basically, what you're looking at are the differences in genes which are expressed on the left. These are the delta EGFR dependent cells versus the ones on the right of the yellow line. These are the delta EGFR independent escaper cells. So you can see a clear difference in color, which each color represents a different gene. The red and the green are genes which are up or down expressed relative to the controls. So there's a set of genes that we found which were specifically regulated in our escaper population, and they're listed here. So we found 19 genes which we're currently in a collaboration with Dr. Bill Hahn at the Dana-Farber, who's helping us screen each one of these genes individually to look at its relevance for growth in the absence of delta EGFR.